Meiosis 1, but I'll just go over it real quick. Remember, interphase is the same. You have the cell's doing its normal cell things in interphase. It's duplicating its DNA to make duplicated chromosomes, which is a chromosome with two sister chromatids. Just like mitosis, that happens before mitosis, it also happens before meiosis. And you can't see the DNA, it exists as decondensed chromatin. Then here in prophase, you have prophase, metaphase, anaphase. In this slide in prophase, the nucleus has dissolved, fragments of the nuclear envelope. You have the homologous pairs crossing over with each other, so pairing up and crossing over. And we call that a tetrad because if one duplicated chromosome is a dyad, di means two, so it's two sister chromatids, it's a dyad. Two duplicated chromosomes put together, the duplicated homologous pairs are called tetrads. So there's 4 sister chromatids total here. If that terminology confuses you, don't worry, I don't use it. I just use duplicated and unduplicated chromosomes. So you have all the chromosomes are duplicated. The homologous pairs find each other. You have crossing over. That causes genetic recombination to occur. Then you have in metaphase, you have the cells lining up on the equator. Again, that's directed by the spindle fibers that were formed in prophase. The homologous pairs get pulled apart, and that's when you can see what's happened. That happened in prophase 1, but you can see the results of genetic recombination. So each of these chromosomes is a mix of the maternal and paternal sets of genetic information. Then you have telophase and cytokinesis, which your book puts them together because they do overlap often. So telophase is when the nucleus reforms, the spindle fibers disassemble, and the chromosomes start decondensing into chromatin again. And cytokinesis is just when there's a cleavage furrow, the cell gets cinched in the middle, the waist gets smaller and smaller and smaller until the 2 cells pinch apart. And now you can see that each of these cells now is haploid. One of each of those 3 chromosomes. All right. I'm not sure why meiosis 1 is there again. It's annoying. Okay. Meiosis 2. You've got the, okay, so prophase 2, same thing, chromosomes, the chromatin condenses into chromosomes, nuclear envelope dissolves, spindle fibers start to form, metaphase 2, line up on the equator. But now you have, remember, these are the 2 that you ended meiosis 1 with, and each of them is haploid. Each one only has 3 chromosomes, and each of them is different, right? So now we're just splitting apart those sister chromatids, the sister chromatids. So now the sister chromatids split apart, and in anaphase, and here in telophase 2 and cytokinesis, you have they decondense, the chromosomes decondense to chromatin, nuclear envelope starts to reform, spindle fibers disassemble, and cytokinesis is the dividing of the cell. And now I don't really like that it combines these because I don't want these chromosomes to be decondensed yet, because I want you to see that each of these daughter cells now, after meiosis 1 and meiosis 2, each of them is genetically unique. So if you were to, if we look at the anaphase one where they're not decondensed yet, and just imagine a line being drawn down the middle here, because that's where those cells will divide. That's the divides the genetic information for each cell. You can see that this set of genetic information is a unique combination of the maternal and paternal genes. This one's unique. No other of these sex cells has this combination. No other of these sex cells has this combination or this combination. So that's why it's important to color code the maternal and paternal sets of genetic information, because that allows you to see that the shuffling that occurred in meiosis 1 is what results in these unique sets of combinations of maternal and paternal genes in each daughter cell. Each daughter cell still has one complete set of genetic information. They'll have one of every eye gene, one of every nose gene, one of every blood gene, one of every gene. So it's a complete set of genetic information, but it's a unique mix of the maternal and paternal sets. So when you're thinking about this, it's hard to, you were thinking about multi-generations here. So the sex cells, this is the end. This is the 4 sex cells. So let's just say they're my eggs. Let's just say they're my eggs. Each of these eggs has a unique combination of my mother's genes and my father's genes. Remember, so I am exactly 50% my mother and 50% my father. But my sex cells, some of my sex cells have more of my father in them and some of them have more of my mother in them. It all depends on how that shuffling happened during meiosis 1. So this one is mostly blue, this one's mostly red. So you can see that, my offspring are not going to be equal parts my father and my mother. It will be, my offspring will be 50% me and 50% my husband, but what they get from me is sort of up to chance. Whether they get more of my father's genes or more of my mother's genes just depends on what happened during meiosis for the particular sex cell that got fertilized. All right, and then in the next little segment of lectures, very short, I'm just going to bring this full circle so you can see how meiosis fits into the full life cycle.